Hello everyone, I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.J. Hariharan. So today we are going to discuss about the bacterial structure internal to the cell wall. Because if you, I have started this series of lecture especially for the structure of the bacteria is that I have divided into three parts. One is bacterial structure external to the cell wall. Second, cell wall of bacteria, and this video I have dedicated to get an in-depth insight about the bacterial structure internal to the cell wall. So let us understand this one by basically starting with what is meant by a bacteria. So the bacteria is an unicellular microorganism which is having a size around zero point 0.5 to 2 micrometer in size, and it can be viewed only under a microscope. It cannot be seen with a naked eye. And moreover, the bacteria is exhibits a typical prokaryotic cell structure. That means prokaryotic means the bacteria doesn't have a well-defined cell membrane, internal cell organelle membrane. So like nucleus which is present in the eukaryotes. So in eukaryotes you see that the DNA is generally packed inside a membrane like structure called as nucleus, which is the DNA is covered with nuclear membrane. Whereas in the prokaryotes like bacteria, the nucleus will be present in the cytoplasm only. It doesn't have a well-bound membrane structure. That means it doesn't have any organelles with a well-bound membrane structure. So if we want to study the structure of the bacteria, generally we can study by dividing its structure into three areas. One is the structure external to the cell wall, second is the bacterial cell wall, and third is the structure internal to the cell wall. So I have already in my channel you can see two videos. One is the structure external to the cell wall, and the second part is the structure cell wall of the bacteria. So this video is totally dedicated about the structure internal to the cell wall. So if we see a simple structure of the bacteria will have the major part, that is the cell wall. Then the immediate structure beneath the cell wall, we can see the plasma membrane. Then it carries cytoplasm. In cytoplasm, you can see ribosomes, which is distributed throughout the cytoplasm. And the DNA is concentrated at a particular area we call as a nucleoid. Then apart from we can having certain inclusion bodies. So if you see the external structure, the major one is the locomotor organ flagellum, which is the long thread-like structure, and a short thread-like structure we can see fimbriae. Apart from this, the covering of the cell wall, it may be a capsule or a layer or slime layer, we can see it. So today's lecture we will discuss elaborately on about the cell structure which is internal to the cell wall. So the various structure which is present internal to the cell wall is that one is the cytoplasmic membrane, which is the major one. And in the cytoplasmic membrane we will get two terminologies we will understand. One is the protoplast and the spheroplast, and the second one is the cytoplasm. And the cytoplasm carries certain cytoplasmic inclusion bodies or vacuoles we can call. Then finally we will discuss about the genetic material, nuclear material, which is present in the cytoplasm. So let us understand with the cytoplasmic membrane. So the cytoplasmic membrane is the immediate membrane which is present beneath the cell wall. So whether it's a gram-positive cell wall, or a gram-positive bacteria, or gram-negative bacteria, the membrane layer which is present just beneath the cell wall is the cytoplasmic membrane. It is having a thickness around 7.5 nanometer generally, but it can vary in the thickness around 5 to 10 nanometer in size. The major functions of the plasma membrane, what is the major role of the plasma membrane, if you see, is the one prime role is that it has to retain the cytoplasm, the liquid portion of the bacteria. The second major function, it acts as a selective permeable membrane barrier. It only allows certain ions and molecules to move inside and outside of the cell. So majorly it maintains the tonicity of the bacteria, and moreover it helps in the transport of the certain nutrient materials like glucose or water molecules inside and outside of the cell membrane, right? Apart from that, it has another major function which is very crucial for its metabolic process. So the plasma membrane acts as a crucial part of the most of the metabolic process of the bacteria, like respiration, photosynthesis if it is a phototropic bacteria, and synthesis of the cell wall membrane, also certain lipid molecule. So apart from that, certain bacteria carries a special receptor-like structures we generally call as a chemoreceptor, which senses the chemicals around the bacteria, whether it is toxic to him or it's a nutrient one. So this chemoreceptors, which is present as a peripheral protein of the plasma membrane, helps to detect these characteristics. So the major role of the cytoplasmic membrane is that to retain the cytoplasm, acts as a permeability barrier, has an important role in the metabolic process, and it has a certain receptors like chemoreceptors on its surface. So to study the cytoplasmic membrane, the most accepted model is the fluid mosaic model. So this fluid mosaic model was developed by Singer and Nicholson, and this model is applied to all the type of cytoplasmic membrane which is in eukaryotes or prokaryotes in nature. So today we will more concentrate on the prokaryotic nature by using this fluid mosaic model. So this model says that the plasma membrane is basically made up of a lipid bilayer. A phospholipid bilayer membrane is present. So in between it, it carries certain protein molecules. This you can see from the picture. So this yellow color layer, what we are seeing is the phospholipid bilayer. So this one is the phospholipid bilayer which is spread throughout the area. Because it is phospholipid bilayer, it has a hydrophilic head and a hydrophobic tail portion. So the hydrophobic tail portion is beneath that because outside water will be present and the inside cytoplasm also hydrophilic in nature. So the hydrophobic one will be inside. This is the phospholipid bilayer. And to maintain the fluidity, it carries certain ring structure like compounds called as hapnoids. That is the like steroid ring structures will be there. So apart from this, it carries certain protein structure which can integrated will be there. We call as an integral protein. Protein like structures will be present. Some will be in outside, which is generally a peripheral protein in nature. Right. So generally, I have already told the structure, that is the thickness of the plasma membrane will be around 5 to 10 nanometer thickness. It is basically made up of a lipid bilayer. So it's a two sheet of molecules. One is a hydrophilic region, another is a hydrophobic region. This is becomes a single sheet, and another sheet will be there. Thereby it forms a lipid bilayer. We can see it. And in between this we can see different type of membrane proteins. Certain globular proteins will be there, certain linear proteins will also be present. So first we have to understand the lipid bilayer, this phospholipid bilayer. So the chemical nature of this lipid phospholipid is that it forms bilayer immediately without the spend of energy. It is a self-assembly bilayer. So if you take a phospholipid and put it into your water, it immediately forms a bilayer. So if you see the structure, it is asymmetric in nature. It carries two regions. One is an hydrophilic region which is in polar in nature. Another is a hydrophobic region which is generally non-polar in nature. That's why since the same structure carries a polar and non-polar group, we call as amphiphatic in nature. So this polar and non-polar is integrated through a glycerol molecule. So if you see a glycerol molecule, so the simple chemical structure of glycerol is CH2OH, CHOH and CH2OH. So one of the CH2OH through phosphate group, it is linked with an hydrophilic region we call as an ethanolamine. So the hydrophilic region may be an ethanolamine or a choline or phosphatidyl inositol, anything can be there. You can see this is the hydrophilic region, right? It is polar in nature, that is water loving in nature. And the second part of the glycerol is connected with a fatty acid group, long chain fatty acids, which may be a malic acid, malonic acid or stearic acid may be nature. So this you can see the two tail like portion. It is hydrophobic in nature because it doesn't likes water. It repels from the water. So it is an amphiphatic molecule carrying a hydrophilic head, which is basically made up of a ethanolamine, choline or phosphatidyl inositol, Majorly ethanolamine will be present in bacteria. This is connected to two fatty acid side chain which is hydrophobic in nature through a glycerol molecule, right? As you can see from this picture, right? So this amphipathic molecule carrying a hydrophilic end and a hydrophobic end. So we call this hydrophilic because it interacts with water. So it forms a bilayer. Out of this two bilayer, the hydrophilic end will be outside or towards the cytoplasmic side. So what happens is that the hydrophilic head will be like this. So two ring, another hydrophilic. So it will be either in water side or an outside or cytoplasmic side. Whereas the hydrophobic ends are insoluble in water, it does not react with water, it interacts with each other. So thereby it forms a immediate bilayer structure, right? So the outer surface is generally hydrophilic in nature, whether it comes in contact with external environment which carries water, otherwise it comes in internal environment which is carries a cytoplasm. So both side of the outer layer of the lipid bilayer will be hydrophilic in nature, whereas the hydrophobic, the fatty acids is get buried inside the membrane. So thereby we can get this structure. So we have this hydrophilic head, another hydrophilic head. So the hydrophobic end is buried inside it. So it interacts and forms the bilayer, because the outer side will be either it carries water or the inner cytoplasmic side it carries the water molecule, right? So this is the measure how the bilayer is formed. So in this plasma membrane, it carries membrane proteins. These proteins are generally acts as a receptors for the transport of molecule or as a receptors for sensing certain chemical entity like chemoreceptors. So generally these membrane proteins are of two types, based on its structure and the ability with interaction with different compounds. One is the peripheral protein, second is the integral proteins. So the name itself says that the peripheral proteins are loosely connected and present on the surface of the membrane, which it generally interacts with the outside chemicals, acts as a receptors. So it constitutes 20 to 30% of the total protein which is present in the plasma membrane. And second is the integral protein. It is generally amphipathic in nature because it carries both hydrophilic amino acid and hydrophobic amino acids. So these hydrophilic regions will be present in the both side, outer side of this plasma membrane, whereas the hydrophobic gets penetrated through the lipid bilayer, because the inner side will be carries fatty acid, so it can easily interact with this. So because of this nature, they can easily diffuse to no new location. So the integral membrane protein can move to any places as they want. It is like a flip-flop or rotating nature it has. And moreover, it carries around 70 to 80% of the total protein. So the third major chemical which is present in the plasma membrane is the Hopanoids. It is a steroid-like structure because if the phospholipid bilayer and membrane proteins only are present, it is more flexible in nature. To give little bit hardness, the sterol, the ring-shaped structure molecules will be needed. So in eukaryotes, if see the cholesterol will be present, so this gives a rigidity to the membrane structures. But in bacteria, this sterol-like similar structure hopanoids will be present. So as you can see from this picture, the bacteriohopanotetrol will help in this activity like similar to cholesterol to give rigidity to the cell membrane. So it carries a sterol ring structure. As you can see, the cycloperhydrophenanthrene ring will be present. So it will help in the rigidity of the cytoplasmic membrane. So for our studies, we have developed certain only cytoplasmic membrane structure with the cell molecule. We call as a protoplast and spheroplast. So let us understand one by one. The first one is the protoplast. So the protoplast is a bacterial portion which lacks cell wall, but it contains only cytoplasmic membrane and the cell material, that is the cytoplasm and the genetic material, everything will be there. That means protoplast is nothing but a bacterial cell which does not have a cell wall. We can prepare this protoplast only from a gram positive bacteria. So the gram-positive bacteria is basically made up of a cell wall, carries a peptidoglycan layer. If you treat with lysozyme, this cell wall get dissolved. So we will get the protoplast, that is cytoplasmic membrane with inside cytoplasm carrying the other material. Or either we can treat with lysozyme, otherwise we can grow the gram-positive bacteria into a media carrying penicillin, because the penicillin acts as a it's an antibiotic. The penicillin is an antibiotic. So if you see that the penicillin generally uh, inhibits the cell wall of the bacteria. So penicillin, by inhibiting the growth of cell wall of the bacteria, we will get protoplast only in an isotonic solution. So the characteristic of the protoplast, you can see the bacteria will be alive in nature, but it will alive only in an isotonic solution. If we put a protoplast bacteria into a hypotonic or hypertonic solution, so in hypotonic, what happen is that it will take the water inside, it will swell and it will bulge and it will destroy it. Otherwise, if we put in hypertonic solution, it loses its water molecule, it shrinks and it will die. So the protoplast bacteria will can survive only in isotonic solution, because the cell wall of the bacteria osmotically protects the bacteria from the hypertonic and hypotonic environment. So whatever the structure of bacteria, so once we remove the cell wall as in protoplast, we will get a spherical shaped bacteria only, right? The second one is the Spheroplast. The Spheroplast is similar to Protoplast. It is an osmotically fragile form, but it is produced in a gram-negative bacteria similar to the Protoplast, because either the penicillin or lysozyme only destroys the peptidoglycan layer. So in gram-negative bacteria, we have two external layers. One is the outer membrane and second is the peptidoglycan layer. So if we add a lysozyme or grow in a penicillin place, what happen is that the peptidoglycan layer will be destroyed and it carries outer membrane. So the Spheroplast will carries only outer membrane of the cell wall. So the Spheroplast is containing an outer membrane, plasma membrane, then the cytoplasm and its organelles, right? So it is, we can simply call as a protoplast with an outer membrane, we call as a Spheroplast. This production of protoplast and Spheroplast is generally done for the study purpose. So it is doesn't naturally occurs in any type of bacterial structure, internal structures, right? So the second major part of the bacterial internal structure is the cytoplasm. So it is the liquid part of the bacteria and most of the organelles are present in the cytoplasm. So the major one is the ribosome, which is granular in nature. Second is the chromatin area, rich in DNA and RNA. And third is a fluid portion, which is generally contains a storage granules. So let us understand one by one. The first one is the ribosomes. So the ribosomes are the major granular part we can see in the cytoplasm of a bacteria. So it is freely distributed in the cytoplasm, whereas in eukaryotes you can see the ribosomes is bound with endoplasmic reticulum, we call as rough endoplasmic reticulum. Some are free in nature in eukaryotic cells, whereas in prokaryotics the ribosomes will be free in nature. This ribosomes is majorly made up of protein and RNA, rRNA, ribosomal RNA. This needs for the protein synthesis. So for the synthesis of proteins, to perform the translation process, the ribosome is needed. So the ribosome, mRNA and tRNA will help in the protein synthesis. So we can differentiate this the type of ribosome of a prokaryotic bacteria and eukaryotic cells based on its sedimentation rate. So we call as an Svedberg unit, its sedimentation. Generally, bacterial ribosomes has two subunits, one large subunit and one small subunit. By together, both the subunits, the sedimentation rate will be 70 years. If the large subunits alone get sedimented, it has 50 years and the smaller one has 30 years. Whereas the eukaryotic ribosomes, the total ribosome sedimentation rate will be 80 years. The larger one will have 60 years and the smaller one will have a 40 years. This is the major difference between the ribosomal structure of the bacteria and the eukaryotes. And the second one is the nucleotide. which is nucleoid, which is present in the bacteria. So as I have already told you that in the location of the genetic material is the striking difference between the prokaryotes and the eukaryotes, because the eukaryotes carries a membrane bound structure, internal structure. That means the DNA is packed inside a nuclear membrane, so that is we call as a nucleus, whereas the prokaryotes doesn't inside a nucleus. It is generally located in the cytoplasm itself. And the second one, if we see that eukaryotes generally carries the DNA is packed in the form of a chromosome. So it is generally 2 or more in number, whereas the prokaryotes will have a single chromosome. And moreover, the it lacks the nucleus. The prokaryote doesn't have a membrane structure like nucleus. And moreover, the prokaryotic chromosome are irregular in nature. Generally, it carries one. So that's why it is called as nucleoid. It is concentrated at a particular location of the cytoplasm. So it is called as nucleoid. Otherwise, we can also call as a nuclear body, chromatin body, or nuclear region. Similar to eukaryotes, the genetic material of the bacteria is the DNA. So the nucleoid carries DNA, that is the double-stranded deoxyribonucleic acid. It is a single chromosome will be present in circular form. Rarely one or two bacteria will have a linear DNA chromosome. For example, if you can see, the Vibrio cholera will have a linear chromosome. Sometimes it have more than one chromosome will be present. So if you isolate this nucleotide and study the chemical nature of this nucleotide, it generally carries around 60% of DNA and 30% of RNA and 10% of protein by weight. So why this nucleotide carries these three different things is that, so the prokaryotes will have a doubling time. That means a single cell can divide into two cells within a short period of time, around 15 to 20 minutes. That's why the various genetic process, that is the replication, transcription, that is formation of RNA from DNA, then translation, synthesis of protein from the RNA, all happen simultaneously, and moreover it is present in the cytoplasm itself. That's why while I take out the nucleotide, the same time the transcription will be happening. So the from DNA, RNA will be present. Same time the translation will be also happening. So the protein also will be present. That's why it reveals 60%, 30%, and 10% of the protein. And moreover, the DNA is condensed with the help of a protein called histone in eukaryotes. So around histone, what happens is the DNA will circle, then it condenses and forms a chromosome. Whereas in prokaryotes, it doesn't contain histone, it contains other protein subunits for wrapping this DNA, right? So this is the major chromosomal genetic material. And another genetic material which is largely present in some kinds of bacteria we call as plasmid. So the plasmid is an extra chromosomal DNA present in a bacteria. So in addition to the normal genetic material, certain prokaryotes carries an additional genetic material. We call it as a plasmid. This is an extra chromosomal DNA molecule. It is generally smaller when compared to chromosomal DNA and circular in nature, and it carries a double stranded DNA. It can multiply on its own. So this unique characteristics helps to develop as a vector for the rDNA technology. In some species, these plasmid are numerous in nature and have different characteristics. For example, in Bacillus burgdorferi, we can carry nearly 12 linear and 9 circular plasmids are present. So the major characteristics of the plasmid itself, it can replicate on its own. It carries an origin of replication initiation. So it can make more and more copies itself. So that's why this plasmid has used as an vector in our DNA technology. And moreover, the plasmids carries only few genes, around 30 genes or less than 30 genes can be seen in plasmid. And more these genes are responsible for conjugation and antibiotic resistance. So these genes can produce pili, in singular we call as a pilus, a hair-like structure which helps in the transfer of genetic material from one bacteria to another bacteria by direct contact. And this process we can call as a conjugation. And generally the conjugative plasmid which carries a fertility factor F factor will favor this conjugation process. So since it can make multiple copies, so we call as a copy number. Certain bacteria will have a single copy of the plasmid. Certain bacteria have more than 40, even 1000 copies of the same plasmid. We call as a multiple copy plasmid. And another terminology we call as an episome. That means certain bacterial plasmid can get integrated with the chromosome. So then we call as an episomes, right? So these episomes sometimes it can come against and can be converted instead of F factor. It can be F prime factor. It can form. So this you can see in my another video lecture regarding conjugation. So the last part of the structural present internal to the cell wall is the cytoplasmic inclusion and the vacuoles. So these cytoplasmic inclusion and vacuoles all generally work as a storage unit for the bacteria. It works as a reserve deposit, a concentrated reserve deposits, and it can be easily detectable with some staining techniques. So one major vacuoles what we can see is the metachromatic granules, also called as volutin granules, which is generally storage for the phosphates. So it can be easily identified with a light microscope once it is stained with methylene blue, dilute methylene blue. Similarly, for the storage of the carbon, it is stored in the form of a PHB granules, that is the polybeta hydroxybutyrate granules for the storage of the carbon. And similarly, the glycogen is stored as a glycogen polysaccharide granules, and it can also be identified in a brown color once it is stained with iodine. Apart from this, elemental sulfur granules are present, and many other storage granules also present. I think this lecture will be might be helpful for you understanding about the in-depth study about the internal structure of the bacteria. So thank you very much. I will surely assure you that you will continually see various lectures regarding the microbiology as well as in the biotechnology and other subjects also in my channel. So thank you very much. Bye.